Yeah, read all of chapter 13, and then many matching parts of 14. No, I'm just going to do a Connect because we're not going to do all of chapter 14, and Connect doesn't give me any way to pick and choose questions. So, yeah, there's just the chapter 13 assignment. All right, I think we're finally up and running here. So, yeah, we're talking about our B cells and our T cells when we left off last Thursday. Really? Does it look normal on the monitor?
Yeah.
Can everybody see? Okay. Okay, so we are talking about our B cells and our T cells, which are the main players in our third lines of defense. And when we left off, let's see, we had talked about how B cells and T cells see antigens, molecules that they're going to respond to. And we had just talked about this. So T cells, unlike the B cells that we're getting ready to talk about more, T cells don't see antigens like on a bacterial cell or a virus or a yeast or whatever. They see those antigens only when they're presented to them, usually by a macrophage or a dendritic cell. So those cells have to digest a microbe first, chop up its antigens, these molecules, into little pieces. They display them on their surfaces inside proteins that are called MHC class II proteins. Those are antigen-presenting proteins. And then here we have a helper T cell. It has a receptor on its surface that attaches to that little antigen fragment. And that will lead to in this case, a helper T cell getting stimulated. It'll divide over and over and over again. Once this has happened, we'll create a little T cell army. And we talked a little bit about how there are different types of helper T cells. Helper T cells are one type of T cell. But then also, really importantly, we talked about how if a T cell gets activated by its antigen, so that cell's going to divide over and over and over again. It'll probably create thousands of copies. And a low percentage of those cells are not going to really do anything right now. They go into a long-lived dormant state. They're called memory T cells. And basically what they're going to do is they're going to wait around in your lymph nodes and spleen and tonsils and places like that. And if you ever get exposed to that same foreign substance again, that same antigen, they get stimulated really, really quickly. They'll make a much faster response the next time around. So those memory cells are really critical for our third lines of defense. This is where our memory comes from in our immune system. Okay, there's another type of T cell. So what we talked about last Thursday were mainly the helper T cells, but there's another type called cytotoxic T cells, and they have a very different job. Your helper T cells, once they get stimulated, their job is actually to secrete chemicals that stimulate other cells of the immune system, including B cells, other T cells, macrophages. They stimulate these other cells to do their jobs better, so that's why they're called helper T cells. It might kind of sound like, well, you could probably get rid of those guys and your immune system would work properly anyway, but that's not true. Your helper T cells are absolutely critical for your immune system to be able to work properly, and that is very evident when we think about AIDS patients, because the cells that they're losing are their helper T cells, and so we can see how poorly the immune system works if you have HIV infection. The cytotoxic T cells, they get stimulated in a similar way to our helper T cell. So this is supposed to be a cytotoxic T cell here. It also has to see the antigen it's going to respond to. It has to get presented to it. So here we've got a macrophage or a dendritic cell that found this virus or bacterium, whatever it is, and digested it and presented the little antigen pieces on its surface. The cytotoxic T cells, these are also called CD8 T cells. That's another name for those because they have CD8 on their surface. That's a receptor or a protein molecule they have on their surface, CD8. Just like we called the helper T cells CD4 T cells, these T cells don't have CD4 on their surface. They have CD8 instead. Also, their T cell receptors attach to little antigen pieces that are inside MHC class I proteins. Helper T cells see antigen pieces inside MHC class II. These guys see MHC class I, like they're showing you here on the diagram. And that becomes really important because not only do your macrophages and your dendritic cells show little antigen pieces on their surfaces in MHC class I proteins, almost all of the cells of our body do that. So if a cell in your kidney becomes infected with a virus, it's going to chop up little pieces of the antigens of that virus, and it's going to show those little pieces on its surface inside these MHC1 proteins. So they do essentially the same thing as MHC class II, but the MHC class I proteins are much more widespread in the body. They exist on almost every cell type that we have. And the reason they're doing that is they're trying to show those antigen pieces. They're trying to attract in these cytotoxic T cells. They're basically saying, come over here and kill me because I'm infected with the virus or I'm infected with a bacterium, and we need to prevent it from spreading elsewhere in the body. Okay, so when this happens, so remember your T cells and your B cells, they generally hang out in your lymph nodes, tonsils, places like that. They're lurking there waiting to see whether they'll ever respond, they'll ever have a chance to even see the antigen they're programmed to react to. If that happens for this CD8 T cell here, that, and notice over here this TH1, that's a helper T cell. It's going to be producing chemicals to help stimulate this cytotoxic T cell we have here. It's going to divide over and over again. That's going to create an army of these guys. I know they're just showing one in the picture, but there would likely be thousands of copies. Those are activated T cells. The activated T cells are then going to leave lymph nodes, spleen, wherever, and they're going to go migrating through the body. They're going to get into the blood, and the blood will carry them to wherever an infection has taken place. So they're actually going to leave the blood and go into, if you have inflammation going on in a tissue, that's going to be the red flag. Exit the blood here and come into the tissue and find the cells in this organ or whatever that are infected with the virus. All right, once they're in the neighborhood where a virus is causing an infection, this is one of your cells that's infected. Here's our T cell. And they're going to get together, like you see here. The little squiggly right there is an MHC class I protein. That's on the infected host cell. It's got a little antigen piece from the virus or whatever has infected it on the surface. Cytotoxic T cell comes along and hooks up with that. And once they make that connection, the T cell, the cytotoxic T cell, secretes substances that cause this host cell to die. It winds up getting destroyed. And those are called perforins and granzymes, which we'll take a closer look at those on the next slide. Also notice you do get memory T cells with these. Again, they're just like the memory T cells we talked about before. They're not going to do anything right now. They're going to wait around and see if you ever get exposed to this same antigen again. And if you do, they're going to get stimulated really, really quickly. They're going to rapidly create an army of activated CD8 T cells, which can then go find the infection wherever it's happening and hopefully knock it out before you even know that it's there. And here is a blow-up. I think I've already been over all of this. This is a blow-up of a cytotoxic T cell getting together with one of your infected host cells. And then the cytotoxic T cell torpedoes that poor little infected host cell. Yeah, this is the one I was looking for. This is another look at what's going on there in a little bit more detail. So this is supposed to be one of your cells that's infected with a virus. And right here in green, that's a class 1 MHC protein on the surface of that infected cell. And that little diamond right there in purple, that's supposed to be an antigen fragment from the virus that has infected that cell. Cytotoxic T cell comes along. There's its T cell receptor. It looks a little different from how your book is drawing the T cell receptor. And that very specifically attaches to that antigen piece. And then the CD8, what it's doing is it's helping to form a bridge between the cytotoxic T cell and that class 1 protein there. Just like that's what CD4 does. CD4 on a helper T cell helps form the bridge between the helper T cell and the class 2 protein. Helps form the connection, little networking that's going on there. Now a little bit more detail, when these two get together, your cytotoxic T cell up here secretes perforins. Perforins are hole-punching proteins. There's another example of hole-punching proteins used by the immune system. That's going to help kill this cell, poking holes in the membrane. On top of that, there are enzymes called granzymes that enter the target cell there. And those set off a chain reaction of events that leads to apoptosis. Apoptosis is cell suicide. The cell kills itself. Or some people say apoptosis. And that includes, there are several things that go on, but one of the things, the target cell there will actually digest its own DNA. That's one of the things that it does in order to kill itself. It'll digest its own DNA. That's one of the things that happens. That's how it winds up committing cell suicide or apoptosis. There is another type of cell that does a similar thing, and they're called NK cells. NK stands for natural killer. And they are actually when you guys were looking at the blood smears last week, you may have occasionally seen lymphocytes that were bigger than the rest of them. They looked just like lymphocytes, but they were quite a bit bigger than the red blood cells. And those may have been natural killer cells. They look like big giant lymphocytes under the microscope. What they do, though, they're actually part of your second line of defense. The natural killer cells aren't looking for specific viruses. They go on the hunt for cells in your body that are showing signs of viral or bacterial infection in a non-specific way. There can be changes on the cell surface that they're looking for. So they do a pretty good job of helping to kill virus-infected cells. Cancer cells, NK cells, are thought to play a role in helping to control cancer in our bodies as well. Okay, so that's our T cell story. How about our B cells? As we saw last week, B cells, they do have receptors on their surface, just like the T cells do. And their receptors are very, very specific for particular antigens, molecules that they're going to be able to attach to and react to. However, B cells are different. So B cell receptors they'll actually directly attach to bacteria, viruses, yeast, protozoans, whatever they are, they attach to soluble antigens. So the antigen is floating through our body fluids. T cells have to have antigens presented to them by our own cells. So that's a main difference between the two. But just like the T cells, your B cell clones, individual B cells, have receptors on their surface that bind to unique antigens. So a single B cell is only going to respond to one antigen, one specific antigen, but we have a big giant library of billions of different B cells that can respond to just about anything we could possibly get exposed to. Okay, and as we're going to see, all right, so the B cell is here. It has B cell receptors, which are Y-shaped. They kind of look like antibodies. And if this is a virus here, if that virus attaches to a B cell receptor, the B cell is actually going to take in that virus or bacterium or pollen molecule, whatever it is. It's going to take it in. It's going to do endocytosis, kind of similar to phagocytosis. It's going to internalize it, chop it up, and it's actually going to display, just like a macrophage or a dendritic cell would do, it's going to display little pieces of that antigen on its surface inside class 2 proteins. And there's an important reason for that. Oh, I forgot I had a picture of the B cell surface here. So we looked at this on last Thursday. So the little Y-shaped molecules, they look like antibodies, and they really are. They're just sitting on the B cell surface. So again, if a virus attaches to that, all of that's actually going to get internalized. The poor little virus is going to get chopped up, and then pieces of its antigens are going to be displayed on the surface inside these class 2. These are class 2 proteins. So there'll be little antigen pieces stuck up here on the surface. So there's a really important reason for that, because what kind of cells did we say are looking for antigen pieces inside class 2? A type of T cell. The helper T cells. The helper T cells are the ones that are looking for little foreign antigen pieces being displayed inside class 2. That's important because this B cell, most B cells, if they just attach to a virus, a bacterium, whatever, that's not enough for them to become fully activated. They also have to get help from a helper T cell. Most of them, not all of them. Some of them can get activated on their own, but usually they need that assistance from a helper T cell. So that story is shown here. So here's a B cell. So this might be sitting in a lymph node somewhere. And here comes a virus. It says micro, but the way they drew that, that's obviously a virus based on its shape. So a virus comes floating into the lymph node. It's been brought in by the lymphatic vessels. And if that attaches over here to the receptor, it's going to get internalized, and then its antigens are going to get displayed up here on the surface like we were just talking about. And the idea there is the little antigen, the B cell is basically saying, hey, look what I bound my antigen, and I'm showing it now on my surface to try to attract a helper T cell to come over and stimulate it. And that's what's blown up here. So this looks very similar to what we saw with when a macrophage or a dendritic cell was stimulating a helper T cell. Here we're seeing a B cell that's trying to attract a helper T cell to get assistance. All right, so for most B cells, you have to have both of these things occur. This has to occur. A B cell has to attach to its antigen, whatever it is that it's programmed to respond to. That's number one. And then number two, that B cell also has to network or hook up with a helper T cell that also responds to that same antigen, that same foreign molecule. Then it becomes activated, like you see here. So that sounds very complicated. It is pretty complex, but there's a safety mechanism behind this. Why does this have to occur before a B cell can get stimulated? And most likely it is to help protect. Autoimmune diseases are bad news. We don't want our lymphocytes responding to our own antigens, even in error. That can lead to big-time problems. So this is kind of a safety mechanism. It's like you've got to flip two switches before your B cells get stimulated. They have to see the antigen they're supposed to respond to, but on top of that, they also have to hook up with a helper T cell that has also seen this same antigen. That helper T cell has already been stimulated by that same antigen. So it's like two switches have to get flipped. If you guys remember last week, we were talking about how your lymphocytes, they're made in the bone marrow initially, and then your T cells go off to the thymus to fully mature. But we generate B cells and T cells that do respond to our own antigens. That probably happens all the time. However, they're supposed to die. They get tested before they get out of the bone marrow or the thymus and get into our circulation. And if they can respond to our own antigens, they're supposed to die. But sometimes they get out, they escape when they're not supposed to. So this little safety mechanism here, let's say a B cell that responds to one of your own antigens gets out of the bone marrow when it's not supposed to. It's very unlikely a helper T cell that also responds to that same antigen is also is also going to get out of your bone marrow and thymus without being detected before that happens. So we've got a nice little built-in safety mechanism here. Doesn't always work because people still get autoimmune diseases, but things would be much worse probably if we didn't have this safety mechanism built in. All right, so here we've got a B cell that has been fully activated. When it does that, it's going to undergo clonal expansion. Again, that means it's going to divide over and over and over again. And I know our diagram here is not showing that one B cell has divided into probably thousands of copies, but it has. Most of those clones go on to become these cells. They change and specialize into what are called plasma cells. And plasma cells make antibodies. That's their big job. They secrete lots and lots and lots and lots of antibodies. Antibodies are, and these little molecules here that look like snowflakes, those are supposed to be antibodies. All they are, they're proteins, and they bind to that same antigen that activated the B cell in the first place. They bind to the exact same thing. That's what they're showing you here on the diagram. Your textbook has the whole diagram together in one piece, but these little things that look like snowflakes represent antibodies. Those antibodies get into your circulation, they're in your body fluids, and they're going to go off and attach to other copies of that same virus or bacterium or yeast or protozoan or whatever it may be, or pollen, whatever it may be that your body needs to clear out. That's your plasma cells. Just like we saw with the T cells, some of your activated B cells become memory B cells. And they don't make any antibodies right now. They just lurk, sometimes for decades, in your lymph nodes, spleen, tonsils, wherever, in your lymphatic tissues. Hang out there, and if they ever do get exposed to this same thing again, they're going to react really, really, really quickly. It takes a while for all of this rigmarole to happen. With T cells getting activated and B cells getting activated, all that takes, there's a delay the first time you get exposed to something that occurs in order for all of these things to happen. The next time you get exposed to it, your memory B cells, your memory T cells kick in so quickly, you really don't have much of a delay. Your third line of defense kicks in almost immediately, and can usually knock something out before it's capable of making us sick. Okay, some of your activated B cells also become what are called regulatory B cells. And we're not going to worry about that. I think that's going beyond a lot of that. To my knowledge, a lot of that's not well understood yet. I don't really think it should even be in your textbook. But regulation, immune regulation, again, that gets into we don't want our immune responses to become too strong and to go on and on and on and on, because that could eventually wind up damaging our own body. So regulatory cells kind of help dial things back and keep it under control when needed. Okay, a little bit more. All right, so an activated B cell divides over and over and over again, and most of its copies become plasma cells. Plasma cells are big antibody producers. They secrete lots and lots of antibodies. These are proteins. They're not cells. The typical antibody diagram that you see looks like this. It's Y-shaped. And then this is what one actually looks like in 3D. So it doesn't always look exactly like a Y. Sometimes the two prongs of the Y may be rotated further apart like you see there. But a typical antibody it's made up of 4 polypeptides, chains of amino acids. And they're held together, like right there, and right there, and right there, by what are called disulfide bonds. This means there are 2 sulfurs holding them together. All right, everything from here down on the antibody, all of this is called the constant regions. And then at the tips, these are called the B regions or the variable regions. All right, what that means is we can make an almost unlimited number of different types of antibodies that bind to an almost unlimited number of different kinds of antigens we get exposed to. But where these antibodies really vary, the variation is occurring here at the tips. Just like we saw with B cell receptors, because the B cell receptors are essentially just antibodies that are sitting in the membrane of the B cells. So it's at the tips where they vary. The constant parts from antibody to antibody look the same. They're essentially identical. All that variation is going to occur there at the tips. So if a virus comes along, it is going to attach right there at the tip. There's the tip on an antibody there. We've got a more realistic representation of what that antibody would look like. And here's another view of that. So they're down here on the bottom. They're blowing up the variable region at the tips. So at the tips, you're going to have a perfect little 3D pocket that is a very specific match for this antigen, whatever it is. That could be a molecule on a virus, a bacterium, a parasitic worm, whatever it may be. The tips of the antibodies have a very specific little pocket. Same thing with your T cell receptors on the T cells. They have a very specific little pocket in them that attaches to a particular kind of antigen. And this binding is so strong and so specific, we can actually use antibodies as tools, which is when we do blood typing on Thursday, we'll see an example of that. When you do blood typing, you're using pre-made antibodies as tools to let you know what your blood type is. It's looking for, what kind of molecules you have on the surfaces of your red blood cells determines your blood type. Same thing with the home pregnancy test. We're not going to get to do that one, but when you do a home pregnancy test, there are antibodies in the home pregnancy test that attach to HCG, the pregnancy hormone. So you can actually, they're used all the time as tools to help us diagnose different things. All right, so how do these antibodies help us? How do they help us clear things that are foreign to the body out of the body? There are several different ways they can do so. They're summarized in this table 13.7 in your textbook. Once you make them, the antibodies are kind of like tags. So the antibodies themselves, they don't actually kill anything by themselves, but what they do is they attract other components of your immune system to come in and help kill the microbes. So in a sense, they're kind of like tags. This is foreign to the body, and we need to destroy it. So one thing they'll do is they'll coat the surface, like you see here. That's called opsonization. And once they do that, okay, so like if that's a bacterial cell and those antibodies attach to a particular antigen, a typical bacterial cell is going to have lots of copies of that molecule, whatever it is, on its surface. So you can have multiple antibodies that attach to the same bacterium, the same virus, whatever it is. Antibodies are very strong magnets for macrophages, like you see here. So you know how we talked about pathogenic bacteria have things like capsules or M proteins, things like that on their surface that prevent macrophages and neutrophils from phagocytosing them. Once they're coated with antibodies, that's no longer true. The macrophage will attach to the antibodies, and the antibodies are attached to the bacterium or the virus or whatever it is. And now the poor little microbes are going to get ingested anyway and destroyed. So I said when we first started talking about the immune system, I mentioned phagocytosis is usually the end result. The immune system, all of these different weapons we have, most of them their end goal is to get these things phagocytosed so that they are digested and cleared out of the body. So antibodies are like magnets for macrophages. Another thing they do, this is called neutralization. Viruses, they're showing you viruses in this picture. Viruses have to attach to your host cell receptors. If they're coated with antibodies, those antibodies can block that from happening. That's called neutralization. So the viruses can't attach to their host cell receptor, so they're not going to be able to infect your cells. That's called neutralization. On top of that, the poor little antibody-coated viruses are going to get phagocytosed. Because antibodies, again, are like magnets for your phagocytes. All right, what you see here in this picture, this is called agglutination. Which is basically a fancy word for clumping. Because antibodies have those Y shapes, or in some cases they have a snowflake shape. We'll see an example of that shortly. They can network multiple copies of the same microbe together and make clumps out of them. So if you have a bacterial infection and there are billions of bacteria in some tissue somewhere, once the antibodies come floating in, they can start to clump together. Because you've got two on a Y-shaped antibody, this little prong here can attach to one copy of the virus or bacterium, and this one can attach to another copy of the same thing. And so the antibodies are really good at clumping things together. And then why is that beneficial? Well, if you have billions of bacteria in a tissue somewhere, imagine if your macrophages had to go around and find each of those individually. If you can clump them together in big clumps of hundreds or thousands, it's beneficial for your macrophage to gobble up 1,000 at one time instead of just one. So it helps you clear them out better. Okay, another thing that antibodies do, when antibodies attach to a foreign cell of some type, like a bacterial cell, a protozoan, whatever it may be, the antibodies attract in the complement proteins. We didn't really talk a whole lot about complement, but those are proteins that are always present in your blood. that can attach to foreign cells and cause them to burst. That's one of the key things they do. So complements or helps out your antibodies with killing things. Another thing, some antibodies are what we call antitoxins. So here you've got a bacterial cell, and it is making a toxin, an exotoxin, like we talked about in Unit 4. And remember how we talked about those? A lot of times the exotoxins are what damage you. It's not the bacteria themselves, but these nasty toxins they produce. Well, you can make antibodies that attach to toxins. Those are called antitoxins. And so they'll coat the toxin and prevent it from doing its damage. So that's another example of neutralization. The toxin has become neutralized. It can no longer, once it's coated with antibodies, the antibodies block it from going off and damaging your cells or tissues. Okay, your activated B cells actually make different kinds of antibodies. They're in different classes, and we're not going to get too heavy-duty into these different classes. But your book has a pretty good table which shows some of the key highlights. Kind of your typical Y-shaped antibody is this, and it is in a class called IgG. Ig stands for immunoglobulin, which is a fancy name for antibody, immunoglobulin. Immuno is referring to your immune system. Antibody proteins are globular. They're kind of ovalish in shape. They look like a glob. So they're also called immunoglobulins. And Ig is immunoglobulin, and IgG is type G. And those are antibodies that have that kind of typical Y shape. And they make up percentage of total antibodies in your serum. Serum is, has anybody ever learned before what the difference is between serum and plasma in your blood? Anybody ever learned that before? Plasma is the liquid part of your blood, and serum is the plasma minus the clotting factors. It's like if you have blood in a tube in a lab, and the blood clots, and you use up the clotting factors in it, and then you spin all the clotted blood cells down to the bottom of the tube. What you have left on top is called serum, because the clotting factors have been depleted. But of all these various kinds of antibodies you may have, 80% of them fall into that category in your body fluids. Notice here on this table, some types of antibodies can cross the placenta and some cannot. The ones that can are going to be beneficial to the fetus, the developing fetus. Where it says fixes complement, that just means some types of antibodies attract in these complement proteins that can cause microbes to burst, and some don't. And where it says FC binds to, the FC portion is the stem of an antibody. That's called FC. It's the stem. Can the stem dock on to phagocytes? So we were talking about how phagocytes, antibodies are like magnets for phagocytes. So that's true for some of these and not others. So those are IgG. There's another type called IgA. And if you look really closely, that is two of your Y-shaped antibodies linked end to end. And we're going to talk a little bit more about those shortly. There's another type called IgM. Those are the ones that look like snowflakes. So if you look closely, those are a pentamer. You have 5 Y-shaped antibodies that are all attached to each other and kind of form that snowflake type shape. Those are actually, all right, so let's say a B cell gets activated. So that B cell is going to make antibodies that attach to a particular antigen. And they generally make lots of IgM types at first, and then later they switch over to IgG. And the IgMs, if you think about it, those are going to be really good at agglutinating, clumping microbes, because you've got 10 different spots on there that can bind to the same microbe. So they're going to be really good at helping to agglutinate or clump microbes together. The IgG ones, though, are smaller, so they're better at getting out of the blood and into your tissues where an infection is taking place, so that's an advantage of that type. IgA ones, we'll talk more about those shortly. IgE type antibodies, we'll wind up talking, that's another, they look Y-shaped, just like your IgG, but they are a little bit different in terms of what they attract. So you can have IgG antibodies binding to the same virus or bacterium or whatever as IgE, but the other weapons of the immune system that IgG antibodies attract versus IgE, what it attracts can differ. We'll talk more about IgE on Thursday because they are involved in allergies. They're also involved in control. Remember we talked about there's a school of thought that allergies occur because your immune system is making a mistake. It thinks it's attacking a parasitic worm when it's really attacking something else. So these IgE antibodies are also important for attacking parasitic worms as well. There's another type called IgD, and those are really just the antibodies that are the B cell receptors. So they're sitting in the membrane of the B cell. They look just like antibodies, but they're not really freely floating around. Picture over here of those, no. Okay, so a little bit more about these IgA antibodies, and again, those are dimers, so you've got two Y-shaped antibodies that are linked together. And that little green squiggly there is called the secretory component. That's something that a lot of IgA antibodies have on them as well. That distinguishes them from other kinds of antibodies. We have these antibodies on our mucous membranes. That's where they exist. So you don't have as much of these in the blood and other types of body fluids. They're mostly on our mucous membrane. So you will have them in your mucus, saliva, your tears. These are also the ones that are present in breast milk, which is also called colostrum. Especially the early breast milk is considered to be colostrum. So these are special kinds of antibodies. That little green squiggly molecule on the diagram called the secretory component, that helps protect these antibodies from getting digested in the infant's or destroyed in the infant's stomach and small intestine. Oh, nice. Okay, we're back. Oh, wow. Okay. Cool. How nice of it to finally kick in and act normal. IgA antibodies in colostrum are not digested in the stomach or the small intestine of the infant. On top of that, they can actually be absorbed through the lining of the small intestine. So these are big molecule. Antibodies are proteins, and we don't normally absorb intact big molecules like that through the intestinal tract. Proteins have to be chopped apart into their individual amino acids before they get absorbed. But these are special. They can actually be, the cells that line the small intestine of an infant are capable of absorbing these intact antibodies, and then they transfer them over into the infant's blood. So that's how the infant winds up picking up these antibodies from the mother's breast milk. So that's a good thing, because if mom is getting exposed to something, then the baby's probably getting exposed to it as well, and the baby's immune system hasn't fully developed yet. It doesn't have any memory cells that allow it to respond to things really, really quickly. And so this is a good way for the baby to pick up some protective antibodies from the mama. Okay, so I've been talking about how these memory cells, these memory B cells, memory T cells, help you respond much more quickly the next time you get exposed to the same foreign substance, whatever it may be. And this is what this graph is showing you. It's primary response versus secondary response. Primary response is your third line of defense. How long does it take for it to react to something foreign to the body? Secondary response is the next time you get exposed to the same thing. And over here on this graph on the left, antibody titer, T-I-T-E-R, is really referring to the concentration of antibodies you have in your body fluids to a particular antigen. So they call it titer instead of concentration. All right, so notice if you, when you first get exposed to, AG stands for antigen, you get exposed to an antigen, there is a latent period. It takes you a while before you start making any antibodies, and it can be several days. And the reason for that is it takes a long time for these T cells to get activated, these B cells to get activated. It takes a while for all that rigmarole to happen. And then finally, when it does, what they're also showing you here on the graph, the red line stands for the IgM type antibodies, the ones that look like snowflakes. So if you notice, there's a little a burst of those at first. And then in light blue, these are the IgG, the more typical Y-shaped antibody types. And those are produced in greater abundance later. So they're attaching to the same antigen. It's just that the class shifts from the ones that kind of look like snowflakes and are really good at clumping things together over to your more typical Y-shaped antibodies. The purple line on top is the sum of the two types of antibodies together. But then notice what happens the next time you get exposed to that same antigen. Those memory cells detect the antigen really quickly, and boom, they get activated really fast. And you immediately start making lots of IgG antibodies, so the type of antibody changes. You also make some IgM, so you have both classes of antibodies being produced pretty abundantly, very quickly. So there's no lag. And over here, during this lag or latent period, this is why you're sick, because it's taken a while for these third lines of defense to kick in. Over here, you don't have that latent period. You start making all this weaponry really quickly. And in most cases, that's going to clear the infection before you even know it's there. Okay, so now that we've been through the third line of defense, again, third line of defense is specific immunity, immunity to specific antigens. Specific immunity can be divided up into different categories. So we have natural categories and we have artificial categories. And natural just means that you acquire the immunity through a natural process. Artificial means it's through a medical process, like a vaccination. Then we have active versus passive immunity. Active immunity means the patient's own immune system has been stimulated. It's the patient's own B cells and T cells are stimulated. You're making your own antibodies. Passive immunity, on the other hand, is where a person receives pre-made antibodies. They've been generated in a human donor or an animal donor, like a goat or a horse, something like that. Then the antibodies get purified, so you're not getting a whole bunch of horse or goat gunk if you receive these antibodies as a medical treatment. Or we're getting better with making antibodies in petri dishes in the laboratory. That's becoming more and more common. So sometimes when people receive pre-made antibodies, those antibodies have been made through a biotechnology type process, which is actually safer, much safer. That's passive immunity. So your own immune system has not been stimulated. You are receiving pre-made antibodies. Or like when an infant receives antibodies from its mother, that's passive immunity. The infant didn't make the antibodies, the mother did. So we're going to passive immunity, like when an infant receives antibodies from its mother, either across the placenta or through breast milk. That's an example of passive immunity for the infant. The infant is becoming immune, hopefully, to a microbe of some type, but the infant didn't make its own immune system has not responded. Okay, there's a table in your textbook which gives some different examples of naturally acquired active. You can put the two together. So we have naturally acquired active immunity and naturally acquired passive immunity. So natural means it has to occur through a natural process. Naturally acquired active immunity is what we've been talking about. This is where you get exposed to something, your own immune system responds. Naturally acquired passive immunity, that means it has to occur through a natural process, but a person is receiving pre-made antibodies. So like when an infant breastfeeds or a fetus in the womb receives antibodies across the placenta, that's a natural process, but it's passive for the infant. Oops. Antibodies don't last forever. If you go back and look at that table that your textbook has about the different classes of antibodies, it mentions on there the half-life of antibodies. They don't stay in your circulation. Half-life is how long it takes for the concentration of a particular kind of antibody to drop by half. That's half-life. So they don't last forever. They last for a few weeks and then they go away. So that means passive immunity, there's no memory with that. You receive these antibodies, they do their job, but eventually they're going to get cleared out and go away. So they don't last forever. Passive immunity is not long-lasting. There's no memory. Active immunity, memory gets triggered. Okay, then how about our artificial processes down here? Artificially acquired active immunity. That's what we call vaccinations. Those are vaccines where you are deliberately injected with antigens and you respond, your third line of defense responds, and then you develop memory to those antigens. So you artificially receive the antigens of a virus or a bacterium that could make you sick without actually getting sick from it. But you develop memory to it just like you had been naturally exposed. So if you ever do get naturally exposed to the evil pathogen, you already have these memory cells that are ready to respond, and they will hopefully prevent you from getting sick. All right, artificially acquired passive immunity. This occurs when a human patient receives pre-made antibodies as a medical procedure. Like if you go out and get bitten by a eastern diamondback rattlesnake, and yes, your own immune system will react to the venom. You will make antibodies to the venom, but you might be dead or severely damaged before that happens. You don't have time for that latent period. It might take days for you to make your own antibodies. So what they do in those cases is they will inject you with huge amounts of pre-made antibodies that can circulate through your body fluids and attach to the rattlesnake venom and neutralize it. They'll block it from being able to damage you. That's artificially acquired passive immunity. And a little bit more about that. Immunizations, the rest of the topic here from chapter 13 is about immunizations. So these are artificially acquired immunity examples. And passive immunizations, this is not what we normally think about when somebody's getting immunized or vaccinated. This is done in emergency type situations. You receive pre-made antibodies. They've been made in an animal, a human donor, or maybe in a laboratory in a petri dish. You receive them in large doses. Here are some examples. This is called CroFab. And that is a mixture of antibodies that will attach to the venoms of, I think it's the 3 most common snakes, venomous snakes in the United States. So it's the rattlesnakes and the copperheads and the cottonmouths. I think the coral snakes are included. I might be wrong about that. I can't read the label on here. I want to say the coral snakes are included as well. And if I remember correctly, it doesn't matter. It'll work for the western rattlesnakes and the eastern rattlesnakes. It works for the copperheads. I'm pretty sure the coral snakes are in there as well. Okay, and then here's another example. Human rabies immunoglobulin. So if you get bitten by a rabid animal, you don't have time to wait around for your own immune system to make its antibodies to help get rid of the rabies virus. So instead, they will inject you with large amounts of pre-made antibodies that will circulate and find the rabies viruses attached to them and help you clear the rabies viruses out of your body before the rabies viruses have enough time to infect your brain and cause you to die. These are sometimes called antitoxins or antivenoms or antivenins. They're given IV, intravenously. Some people can get sick from these because there are some types that are made in sheep and horses and so forth. And remember, sheep and horse molecules are going to be seen as foreign by our immune system. They're not going to be identical. And so sometimes people have reactions to these antisera. Sometimes they're called antisera. That's another. Antiserum is singular. Antisera is plural. Especially, let's say you get bitten by a rattlesnake and you receive antivenin against the rattlesnake. You get this Crofab stuff over here. I don't remember offhand which type of animal that's made in. And then a couple of years later, you get bitten by a rattlesnake again, and you have to receive these antibodies again. The first time you receive those, you're receiving these animal antibodies. You may not have a strong reaction to it, but you are generating memory of your own to these animal molecules you were injected with. So the next time if you have to receive those again, you're much more likely to have a strong reaction to it. So try not to get bitten by rattlesnakes and black widows and things like that more than once. Rabies, try not to get rabies more than once. That would not be a... Rabies is bad news. I know this was like a couple of years ago. There was a case out in California. There was a little girl who had rabies. I think she was a little girl. I might be wrong about that. I might be getting this mixed up with the brain eating amoebas. I know occasionally there are little kids who have survived those. But somebody out in California survived a natural rabies infection, actually recovered from it, which is almost unheard of. Rabies is, if you don't get treated for it, is almost 100% fatal. Okay, what we typically call vaccines are active immunizations. This is much more common. We all get these. In this case, you're not getting antibodies. You are receiving antigens. So the idea there, you're receiving them artificially. The idea there is to stimulate your own immune system, and we want it to lead to memory. So we're mimicking a natural infection, but you're not becoming sick from it. Vaccines are subjected to a lot of safety testing. It's very extensive. So in spite of all of the hocus pocus we hear out in the news and from people like Jenny McCarthy, vaccinations are safe. I mean, there are pros and cons. I mean, you're being injected with a substance that's not natural to your body. And yes, you might have a mild reaction to that. And sometimes there have been cases where people have strong reactions to it. But the pros outweigh the cons. Many, 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 many more people benefit from vaccinations than are harmed by them. So they don't cause autism. That's a bunch of hocus pocus. The scientist who published a paper linking autism to vaccinations, that has been demonstrated to have been a big-time fraud. He was trying to make a name for himself and made up a bunch of data about that. But unfortunately, this idea has caught on. A lot of people are scared and think that vaccines are responsible for autism when they're not. Okay, so your vaccines need to be safe. Some ideal characteristics of vaccines. Ideally, a vaccine should not require a lot of booster shots where you have to go back over and over again. Unfortunately, that's not always the case. It's the nature of the vaccine.
What are some of the boosters that could we MMR is that?
I think so. A lot of them you do, but then some of them you need multiple boosters, like a tetanus shot. You have to have that. And it's because the ones for which you have to get a lot of boosters, you don't generate, the memory is not as long lasting. You don't stimulate as many arms of your immune system. Like the really good vaccines will stimulate B cells and T cells, and you'll have all these memory helper T cells and memory cytotoxic T cells and memory B cells. Those types are really good. A lot of times you don't even need a booster for those. But then some of the others, like the tetanus.
That we need as an adult, is that
I think offhand that may be. You did have to get an MMR. Yeah. Is that because you're going into nursing school or? Yeah. So some things like that, if you're going to be in a high-risk profession where you might get exposed to people who have those diseases, they probably want you to have a booster. Because for a lot of things, like when we get these vaccines as kids, we still have memory, but the memory gets weaker and weaker and weaker as we get older. And that's probably okay as long as you're not going to be exposed to a lot of kids or whatever who might wind up with measles or mumps or something like that. But if you do work in the healthcare profession, your risk is a little bit higher, so they probably want you to go ahead and have a booster for that. However, unfortunately, with the anti-vaccination movement having been growing, we're starting to see more. It's kind of like what we talked about with whooping cough. With mumps, there's a measles outbreak somewhere in the United States within the last couple of weeks. I think it was in Texas, if I remember correctly. And those types of things put those of us who are middle-aged and older at more risk of getting those childhood diseases because our memory from our vaccinations is going back down. So who knows? Coming down the road, maybe more of us will have to go back and get booster shots for some of these things when we didn't have to before. Okay, vaccines come in different categories, and some of them are called whole cell or virus vaccines, because remember, viruses are not cells. This is where you are getting you're being injected with whole bacterial cells, whole viruses. So the antigens of the bad form of the virus or the live form of the cell or whatever are there. Your immune system detects them and responds to them just like you were being naturally infected. And some of these are called killed cell or inactivated virus vaccines. So you're getting the whole microbe, but it's been killed. I know one of the things that you hear about from the anti-vaccination people is that, the vaccines have formaldehyde in them. Formaldehyde is toxic. Well, they do use formaldehyde for some of these vaccines because that will kill the bacteria or it'll inactivate a virus. And formaldehyde is a preservative. It preserves the three-dimensional structure, which is important because your B cells and your T cells need to see those 3D structures. They need to be preserved. But they're not injecting you with formaldehyde. They use the formaldehyde to pickle the bacteria or the viruses, and then they get rid of the formaldehyde. If there's any left, it's just there in little minute amounts. The other type are called live attenuated cells or viruses, attenuated type vaccines. And so here they're giving you a live microbe. This is like with the Tamiflu when they inject that up your nose. Yeah, the oral polio, we don't give that in the United States anymore, but I think all of us were squirted with the oral poliovirus vaccine. They were giving you attenuated live polio viruses. Polio actually, even though polio naturally infects cells in the small intestine, that's where it goes first. And if you get a natural polio infection in a minority of kids who were infected with polioviruses migrated from the intestinal tract to the spinal cord and infected neurons in the spinal cord, and that's what led to paralysis. That actually happened in a minority of poliovirus infections. The attenuated virus doesn't do that. It goes to your intestinal tract, so it's going to stimulate your immunity. You're going to develop memory to polio. But it doesn't migrate to the spinal cord and kill your motor neurons like it does in the case of the natural infection. However, having said that, it is we were getting live polioviruses, and they multiply in your intestinal tract for a while, so they do mutate. And every year there were a few kids who had mutations of this attenuated polio, and the viruses regained their ability to infect the motor neurons in our spinal cords, and they would wind up with real full-blown poliovirus from it. So that's a case where you got the pros outweighed it. Once we started using that vaccine, the number of cases of poliovirus in the country went down to almost zero. But there were a few kids every year who actually got polio from the vaccine. Once polio was eliminated completely from the United States, we stopped giving the oral poliovirus vaccine. We've gone back to the injected type, and the injected type is an inactivated virus vaccine. So you're getting the poliovirus that has its 3D shape, but it's not multiplying inside you. There's no possibility at all of getting poliovirus from it because it's been inactivated. So you have to weigh the pros and the cons with these different types of vaccinations. There's a worldwide effort to completely, smallpox was completely eliminated from the globe, and there are no natural smallpox infections anymore. It's gone. Smallpox is sitting around in some freezers at Fort Detrick in Maryland, and the Russians have it as well. So it's kind of like the nuclear missiles. We're not going to give up ours until they give up theirs. And there's concern that terrorists, I think we've talked about this before, there's concern that terrorists might get their hands on it and launch a new smallpox outbreak. But once the smallpox was gone from where humans are its only host, so once enough people around the world had been vaccinated, it had no more had no more hosts to infect. We were able to stop vaccinations, so that happened in the early 70s. So they're trying to do, the World Health Organization has been trying for the last 20 years to do this with polio, but they just have not been able to get all the kids around the world vaccinated, so there are still outbreaks from it. Did y'all hear about in Syria? There, with all the civil war there, Syria was a place where there were no natural cases of polio, but it's back because of the unsanitary conditions. Polio is spread fecal-oral because the main place it replicates is in the small intestine. It gets shed in the feces, and so people pick it up from fecal contamination. But there are usually some outbreaks in other parts of the Middle East and in Africa. And we have problems in some communities in those parts of the world. People are distrustful. They don't trust the World Health Organization workers coming in. They think there's a conspiracy to try to inject their kids with poisons and things like that. So that has also limited the ability to wipe poliovirus out, unfortunately. So I'm not sure we're ever going to be able to completely get rid of that one. Okay, another type of vaccine. These are called subunit vaccines. And this is where you're not getting the whole microbe. You're just getting an antigen or antigens. So you might just be injected with the proteins of a virus. And that's okay. Your B cells will still see this. And these antigens get ingested by macrophages. They'll present them to T cells, so you can get T cell immunity from this as well. but they're generally not as good. When you have like a live virus or bacterium multiplying inside you for a little while before you clear it out, that leads to a lot better memory than these types of vaccines. Some examples of these: the pneumococcus vaccine that kids get to prevent. No, pneumococcus is Streptococcus pneumoniae, which can cause meningitis and also can cause pneumonia. Hib is Haemophilus influenzae type B. That's another one that kids get because that can cause meningitis. The hepatitis B vaccine you have to get if you're going into healthcare. You're not getting, we don't have, in a lot of cases, we don't have an attenuated version of the virus that's safe to give to people. You have to think about the safety testing you have to do on something like that if you're going to inject people with a live microbe. And in a lot of cases, we just don't have strains available that are incapable of causing disease. So the hepatitis B is one of those types. So they just give you antigens. So you're just getting pieces of the microbe, so there's absolutely no chance you're going to get sick from that. The pertussis vaccine is like that as well. That's not actually true. There's a wholesale version of the pertussis vaccine. That's been was the traditional one that's used, and now we have subunit vaccines that are being used, I think, as a replacement for the older version. Pertussis has been mutating and becoming resistant to the traditional vaccine. And that, in combination with people not getting their kids vaccinated, has been leading to a lot more cases of pertussis. Another type of vaccine are called toxoid vaccines. And what a toxoid is, so we talked about all these exotoxins that some types of bacteria make. And again, those are often what actually make us sick. A toxoid is a toxin that has been modified. It's been chemically changed. So it's no longer capable of damaging you, but it still has enough of its 3D shape that it will stimulate immunity to the toxin. So some good examples: the tetanus and the diphtheria toxoid vaccinations. These generally don't stimulate very strong immunity, so these are things you generally need boosters for. But like with diphtheria, it's the diphtheria toxin is what makes you sick and can cause that yucky pseudomembrane to develop in the back of the throat and used to suffocate kids. So if you take the diphtheria vaccination, you will make antibodies that will attach to that toxin and neutralize it, basically soak it up, so that it's no longer capable of causing that damage if you actually wind up getting exposed to the diphtheria bacteria. Same thing with tetanus. Tetanus freezes your muscles in place, causes lockjaw. And so the tetanus is the problem. So if you can make antibodies that attach to the tetanus toxin, that can prevent that from happening. So they call that tetanus toxoid vaccine. The diphtheria is part of the DTaP vaccination that kids get, which is diphtheria, tetanus, and pertussis. Adjuvant here. If you guys ever see this on a box or on a vial of a vaccine, it may say it's adjuvanted. Adjuvants, that's kind of a catch-all term. That is additives that are mixed together with the antigens, whatever those are, to help preserve the vaccine. Some of them help prevent the antigens from sticking to the vial, or some of them are included to irritate the injection site, stimulate inflammation more strongly, because that can actually lead to better memory. It leads to a better response. That's why you can get injected and you get a real sore spot. A lot of times that is due to some of these additives they've put in. That's another thing the anti-vaccination crowd gets upset over, are these additional additives that are in there. But they're in there to help make the vaccine work better and hopefully lead to a stronger response. Okay, so when we hear this term tetanus shot, There are different kinds of tetanus shots. So you can, some of these are traditional vaccinations that lead to artificially acquired active immunity. So your own immune system gets stimulated, you develop immunity. And other tetanus shots are passive immunizations. So let's say you step on a rusty nail, and a few weeks later, you have symptoms of lockjaw. So when you go to the doctor's office, and you don't have time to wait around for your own immune system to start responding to the tetanus. Yes, you'll make antibodies to the tetanus toxin of your own that will eventually neutralize it if you don't die first. You don't have time to wait around for that. So in that case, the doctor is going to give you a passive immunization. They're going to load you up with lots of pre-made anti-tetanus toxin antibodies. And those will hopefully neutralize the tetanus toxin. This is obviously an old painting of somebody who had full-blown tetanus before we had tetanus immunizations available. So all of your muscles eventually spasm and you're just kind of locked into a contracted state, and you suffocate because your diaphragm gets locked into a contracted state as well. Now, when you get the DTaP shot, there you're receiving the toxoid, the tetanus toxoid, as part of that vaccination. And that is, or if you get a tetanus booster shot, they're giving that to you to stimulate your own immune system to make its own antibodies. So if you do have a cut or something like that, the doctor says, when was your last tetanus booster? In that case, they're just going to go ahead and give you a regular booster immunization. That's to make sure that you get exposed to enough of this toxoid so that you will re-stimulate your immunity. That way, if you have a little bit of the Clostridium tetani bacteria in a wound somewhere, it's going to take them a while to start cranking out the toxin in large enough amounts to make you sick. So hopefully by that time you have reprimed your immune system to respond on its own. Okay, herd immunity. We'll pick up here. This is a really important topic, so I want to spend a little bit more time on that on Thursday. And I'm going to post Chapter 14 info, and I'll post that either today or tomorrow. And that involves immune disorders, and so we will not be able to cover the whole chapter, so I'm going to pick and choose some things that we'll talk about on Thursday, and that will finish out the immune system. That includes things like we'll spend some time on allergies, since most of us have allergy problems of some type. And we'll spend a little time on autoimmune diseases and immune deficiencies. So we'll talk a little bit more about HIV/AIDS. And that'll be it, and we'll do our blood typing on Thursday. And hopefully we won't have any more weather issues today.